Hello everyone, welcome, and today we will discuss identification and authentication of herbal materials. Earlier session we saw what are herbs. These herbs are part of which kind of formulations, how are they appearing commercially in the market. Now let us see what and how do we identify a particular herb when we want to make a particular formulation for therapeutic use. Herbal materials are plant derived substances which include different plant parts. It can be leaves, roots, stems, flowers, seeds, the stamens, or their extract, or the entire herbal plant itself, and these are used for therapeutic purposes or they are used for health promoting properties as wellness formulations, nutraceuticals, food supplements, dietary supplements. Hence, there is a need for correct identification and authentication of these herbs which become part of a formulation. Now, this identification and authentication ensures the correct use of plant. Botanical misidentification can lead to use of wrong species or plant part, and some of these herbs which look alike can be toxic. For example, Atropa belladonna versus Withania somnifera. Accurate identification ensures therapeutic consistency and avoids adverse effects. This also supports standardization and quality control. Authentication is the first step in standardization of herbal raw material. It ensures that bioactive constituents are present in expected quantity and quality. This also prevents adulteration with substandard or inherent material. Identification and authentication makes sure that we meet all the regulatory requirements as per the pharmacopoeial requirement. The regulatory bodies like Ayush, WHO, US FDA has some mandates for botanical authentication. We cannot start any herbal drug research without a proper authentication certificate. In fact, some of the good journals, when we want to publish literature or scientific proof of a herbal drug, insist on an authentication certificate. Pharmacopoeias specify the macroscopic, microscopic and phytochemical parameters for identity of a particular herb. This ensures compliance with good agricultural and collection practices and also with the good manufacturing practices mentioned today by Ayush and by Government of India. This protects the consumer safety. It prevents any toxic reaction due to substitution or contamination. A simple example is of dill. Dill apiole is a toxic constituent which is present in Indian dill, and the European dill, which is Anethum graveolens, has less amount of dill apiole. So, identifying whether it is Anethum graveolens or Anethum sova becomes very important to recognize the toxic constituent present. This reduces the risk of drug herb interaction by ensuring correct identity of the herb. It builds a consumer trust in the herbal products. It also facilitates research and clinical validation. Accurate identification is foundational for pharmacological studies and clinical trials. It enables reproducibility of results and comparative studies across regions and species. All these herbs are obtained from nature, hence there can be slight variation. So, identification and authentication becomes very important for all these herbs when we collect it from wild sources and across the globe. Suppose they are coming from different habitats, identifying the correct form of the herb is extremely important to deliver the right formulation. It preserves the ethnobotanical knowledge and biodiversity. This helps us document and validate traditional uses of the plant. It prevents biopiracy and supports intellectual property rights through proper documentation. It encourages sustainable use of medicinal flora. Why is authentication of herbal drugs a mandatory requirement when we are doing herbal drug research or herbal drug formulation? Because authentication of a herbal drug is the cornerstone of quality, safety and efficacy of herbal medicine. This is a critical process which ensures that correct plant species and appropriate plant parts are utilized in herbal preparations, protecting both the patients and practitioners from potentially harmful consequences which can be very drastic. It is a process of confirming the genuineness and purity of that identified material, ensuring it is right and unadulterated source. Unlike the synthetic counterparts, here our herbs are packed by nature with different kind of ingredients, and during standardization we try to identify just one compound which may be present, but it is very important to identify the right herb where we can ensure that the other toxic constituents are not present. Specific verification can be controlled which confirms the correct botanical and plant part are sourced and processed. It prevents adverse reaction from misidentified and contaminated plant materials. Authentication ensures quality assurance, maintaining therapeutic efficacy and protects millions of consumers globally. The objective of herb drug identification is to confirm the correct source, the botanical origin, that the herb is collected from a correct habitat, ensuring the minimum amount of active constituent present in a herb which are responsible for the therapeutic activity. It also ensures consistency and quality of a raw herbal material, and it provides a scientific basis for standardization of herbal drug. A crucial role of identification and authentication hence is ensuring safety and efficacy, prevention of adulteration and substitution, supporting quality control and standardization, maintaining the regulatory and commercial importance of a drug, and for scientific validation of traditional knowledge. How is authentication and identification different? Many a times these are taken as synonyms and everyone thinks that we can use these terms alternatively. Identification is the preliminary recognition of a plant. Authentication is verifying that identification. So, the purpose of an identification is to recognize and name the material. So, by looking at the morphological features, we may consider that the particular herb or the plant part is XYZ or can be named. Authentication further verifies it, so we can actually stamp that material and say that this material is genuine, not substituted, adulterated or contaminated. So, when you have a group of a large batch of this material, when you look at the material and ensure that the entire batch by random sampling contains only that herb, then this is said to be authentified. When we look at a small batch and identify it, that restricts us to say that the the herb is only identified and not properly authenticated. So, when we say that a herb is authenticated, it should be put on paper with a proper signature saying that so and so drug is authenticated, and an authentication number is given with a certificate. So, the focus for identification is what is it? It may be so and so, whereas in authentication we claim what the drug is to be. We ascertain and positively claim that this herb is so and so by looking at much more than the morphology. The outcome here is, in identification we label the morphological parts. Here it is verified and the quality is ascertained for safe use. In identification, visual inspection is done, whereas in authentication there is a comparison with authenticated reference. Most of the times, majorly it is compared with a herbarium sheet where the authenticated drug in prior cases is attached there explaining each part of the drug. So, an herbarium sheet has different parts of the plant with the person's name who has collected it and the natural habitat from where it is collected. The microscopic method for identification will look at common features which are present. For example, when we are identifying a bark, whether the cork is present, whether a medullary ray is present, whether secondary phloem is present, then we can identify the powder as a bark. But in case of authentication, suppose we are authenticating cinchona powder, we have to see whether along with cork, medullary rays, there are phloem fibers present. The length of the phloem fibers is so long that it goes out of the eyepiece length. So, when we measure such long phloem fibers, then we can say that the powder is cinchona. As compared to cinnamon, when you are seeing cinnamon powders, you can additionally see stone cells and shorter phloem fibers. So, when we are authenticating, we actually count the length of the phloem fiber and we see for other diagnostic characteristics which ascertain the presence of that particular herb. Chromatographic technique, TLC, HPTLC, HPLC can be used to identify marker compounds. So, most of the times a common marker compound is used to identify, but suppose we use a particular marker. For example, when we are doing authentication of cinchona bark, if we use quinine instead of a tannin or gallic acid, we can ascertain that, or we can authenticate that it is cinchona bark. Or a modern method of authentication is doing a DNA fingerprinting for that particular bark. So, the challenge is to identify adulteration which can lead to misidentification. The herbal medicine industry faces this as a biggest significant challenge, and the reputation of all herbal formulation is at stake because of this misidentification or the adulteration or substitution which is present along with the official drugs. So, even experienced botanists and pharmacognoscists struggle with identification when plants share similar physical characteristics. It is more difficult in case of powdered or processed materials. When processed or powdered material is supplied or is present in the market, it is very difficult to identify the correct authenticated drug, and global survey from this reference says that about 27% of the commercial herbal products were found to be adulterated either with a similar looking drug or with something else. And a study of raw herbal trade in South India showed that 12% of the market samples were found to be adulterated with substitutes. So, maybe not toxic materials, but a substitute has slightly lower amount of active constituent or little different active constituents. Hence, identification and authentication becomes very important, and this should be performed on the raw material before it enters the formulation or before it even subjected to extraction. The most common lab available method for identification is organoleptic, macroscopic, microscopic, physicochemical, and the modern tool identification using chromatographic and spectroscopic, or the DNA fingerprinting, which is called the molecular method of identification. The simplest method of organoleptic evaluation which we use in our daily lives. So, suppose we want to buy a bunch of coriander, fenugreek, or we want to select the best mangoes. Immediately we look at the color, we smell, we taste, and we can identify a good quality day to day vegetables and fruits. So, these same techniques can be applied to authentication of any herbal raw material. So, today if we tell a layman to identify a rose flower with a jasmine flower, by just smelling the flowers we will be able to make out. So, this smell becomes a major important characteristic for us to identify crude drugs also. Organoleptic, here organo is organ and leptic is senses. So, what impression the herb leaves on 5 sense organs is a way to identify or authenticate the material. So, by using your eyes, nose, tongue, the skin and your ears, we can smell, see, taste or touch a herb, and depending on this we can identify a herb. So, for example, recognizing ginger rhizome, dried ginger or fresh ginger by its beige color and wrinkled surface. So, when you look at ginger, if you have observed ginger carefully, a ginger piece, it branches in all directions. This is called symploidal branching. So, you have a branch coming this way, going straight, going right, going down. This is called symploidal branching. Another type is monopoidal branching, where it branches only in one direction. So, these kind of morphological characteristics help us identify a crude drug. Recognizing ginger rhizome. How do you recognize a rhizome? There are many scales, there are buds present, and hence we can identify different kinds of ginger. In ginger, the quality of ginger you can make out by simply smelling it. A strong smell tells us that the active constituents, the oleoresins which are present in ginger, which is shagol and gingerol, whether they are present or not. So, these are some basic organoleptic evaluation which you can do on the crude drug. For example, identifying peppermint leaf by smell. So, here we are using nose as the sense organ to detect the herb. Here we are using eyes to see whether ginger is branching, what is the color. By tasting, you can identify neem leaves. If you have tasted, they are very bitter in taste. So, distinguishing or identifying neem leaves from other leaves by bitter taste, and here feeling the fibrous texture of licorice. If you have ever chewed licorice, when you try to break it and feel the surface, you have lot of fibers coming out from the broken surface. That is a fibrous texture. When you try to break a cinnamon, it has a sharp sound. So, that is the sound which you hear with your ears when you crack, suppose you break a coconut, you have a sharp sound. That sound will help you identify, that is called the crackling sound of each herb, which will help us identify a herb. Looking at the macroscopic visual examination, whether the plant part is whole, it is a cut plant part. So, we can make out whether it is a leaf, a root, a stem, a flower or a fruit. So, from childhood we all know how a leaf looks. It has a lamina, it has a small petiole, it has an apex, it has venation. So, this will help us identify whether it is a leaf. For a root, we know that it is an underground plant part. So, we can differentiate a leaf and a root. A leaf 90% of the time looks green because it has chlorophyll in it. We know how a stem looks. A stem will have nodes, internodes, leaf, branches. So, we can easily identify it from a leaf and a root. A flower has petals, stamens, pollen grains. So, we can identify flower from leaf and root, and a fruit has seeds, it is formed from a flower. So, all these are macroscopic evaluations which unknowingly we are studying right from our childhood. These are external features which help us to identify a crude drug. So, when I say cinnamon and when I say fennel, cinnamon is a bark. Cinnamon is a spice which we use in our kitchen, and fennel is an entire tiny fruit. This is an umbelliferous fruit. So, we know how to identify these fruits from a bark. When I say ginger, this is a rhizome. Since it shows presence of scales and buds, it is not a root, it is a rhizome. So, the size, smell, color will help us identify ginger from say tulsi leaves. Now, if I tell you to imagine a tulsi leaf and a mango leaf, a tulsi leaf is a small leaf, a mango leaf is a huge leaf. If you remember seeing a bay leaf, the lamina of bay leaf is divided into 3 parts. So, thus macroscopic characteristics will help us identify what kind of leaf it is, or whether it is a leaf, a fruit or a stem. For example, bale, as I said, is divided into 3. If we look at durva, it is a sharp grass and there are 3 leaves present like this. That is how we identify durva. If I say a rose leaf, it has serrated margin like this. A mango leaf will have it's a bigger leaf, and as compared to the rose leaf, this margin is entire. If I say datura, the margin has 3 lobes like this. So, with the help of these kind of botanical markers, we will be able to identify a herb. If you look at the aloe leaf which is seen here, aloe leaf has a lot of thorns on the sides. So, we can identify leaves based on how they look. The most simple one for macroscopic examination is whether it is a simple leaf or a compound leaf. A simple leaf is directly attached to the branch like this, whereas a compound leaf, the lamina is divided into many leaflets like this. So, if you see a tamarind leaf or a senna leaf, you will see leaflets like this. So, this is a compound leaf and this is a simple leaf. If you look at the venation, this is a reticulate venation where you can see network of veins. So, when you hold this leaf against light, you will see lot of networks. This is reticulate venation. If you see a banana or a coconut leaf, you will see veins going parallel like this. So, this is parallel venation. You have palmate venation like this, where the venation is the shape of palm. Suppose the main vein comes from this, you have others going, other main midribs or the veins like this. This is called palmate venation. So, by looking at these characteristics, we will be able to identify a particular leaf, or there are floral formulas given, whether a flower has 4 petals, 5 petals, how many stamens, whether the position of the gynoecium is lower as compared to the other floral members. So, these are some of the morphological characteristics which help us identify a herb. Now, if you look at this cinnamon, this is Sri Lankan or Indian cinnamon. You can see the coils here. Both the surfaces are coiled inside. So, it looks, when you look at the surface section, it is like this, and inside also it is packed. But if you look at Indian cassia bark, which is used as a cinnamon, it is a single channeled bark like this. The cross section is like this. So, this helps us identify different barks. Microscopic examination, what we observe under the microscope, this is very much needed for powders. Now, if you look at senna leaf here, senna shows parasitic stomata. What does parasitic stomata means? The guard cell and the epidermal cell, the axis is parallel to each other. So, if this is the guard cell, the epidermal cell in case of senna appears like this. So, this axis and this axis is parallel. We can also see a warty trichome in case of senna. So, this is a unicellular trichome where the cell wall has warty thickenings. And another very important characteristic of senna is the xylem vessel is covered with parenchyma cells, and each parenchyma cell has got a calcium oxalate crystal. This is called the crystal sheath. So, this crystal sheath is seen in case of senna and in case of licorice. But if we see a stomata and a trichome with this, we can say that this is powder of a leaf. If we see cork along with this, we can say it is licorice powder. So, this is how we can identify whether it is a leaf powder, whether it is a bark powder or a fruit powder. Suppose we see endosperm which is rich in oil. This is how you will see an endosperm. Then we can say the powder contains fruit. When we are seeing typical cells of an endosperm, it shows an oil globule with protein and carbohydrates. This is a typical characteristic of an endosperm. Then we can say that the powder contains some kind of fruit or seed. When a powder contains stomata and trichome, we can say that a leaf is present in the powder. Suppose there is cork present, then the powder contains a stem or a root along with presence of xylem or phloem fibers. Quantitative microscopy helps us to identify different species. So, stomatal number, where we count the number of stomatas present in this tiny square of 1 square millimeter, we can identify digitalis lanata and digitalis purpurea. Stomatal index, the ratio of stomata to the total number of epidermal cells, will also help us identify two different leaf species. Quantitative microscopy, the vein eyelet number of vein eyelets. What is a vein islet? An area on the green lamina of the leaf which is covered on all sides by veins or veinlets is called a vein islet. So, counting the number like the stomata will help us identify two different species. So, different species of Cinchona, Cinchona officinalis, Cinchona succirubra, Cinchona calicaya, we can identify with the help of vein islet and vein termination looking at the leaf. Also two different types of senna, the Indian and European senna. Senna Alexandria, which is the European senna, and Senna angustifolia, which is the Indian senna. Palisade ratio, the number of palisade cells to an epidermal cell will also help us identify. Lycopodium spores can be used as a preliminary standard to count the number of diagnostic microscopic characteristics present. Qualitative microscopy is the kind of epidermal cells, the kind of stomata, the kind of trichomes which we discussed earlier can be used to identify. The stomata types, these are different types of stomata. So, depending on the alignment of the epidermal cell with the guard cell, here you can see anisocytic, there are 3 epidermal cells and 1 cell is smaller in comparison. This is anisocytic or cruciferous type of stomata which we see in Datura stramonium, dicytic which we saw in Senna, parasitic which we saw in Senna, and this is irregular shaped cells surrounding the guard cell. So, we can identify a herb depending on the kind of stomata, depending on the kind of trichome, whether it is unicellular, whether it is multicellular. This is trichome of digitalis where one cell in the center is collapsed. This is multicellular covering trichome, specially present in case of digitalis. But such type of trichomes are these days seen even in Datura. So, how do we identify Datura and digitalis? Digitalis has a glandular trichome with a stalk. So, you can see such kind of glandular trichome in case of digitalis. Vasica has a sessile glandular trichome like this. So, we can identify two different types of leaves depending on the kind of trichome, which whether it is unicellular, multicellular or covering. Different kind of calcium oxalate crystals are present. For example, if we look at kurchi bark, you put a pinch of kurchi bark under the microscope, you can see these tiny sugar like crystals. These are prism shaped calcium oxalate crystal. Even without processing kurchi powder, just take a pinch of kurchi powder, put it on the slide and see under the microscope. On the slide, in between different drug particles, you will see the shining white particles. These are calcium oxalate prisms which help us identify that the given powder may be kurchi along with other diagnostic characteristics. If you see rosette type of calcium oxalate crystals, small tiny earring like stud like crystals, this is very distinctly seen in rhubarb powder. Rhubarb rhizome powder, if you see, we can see these crystals, or if you can see needle like crystals, this is seen in squill. So, by looking at the kind of calcium oxalate crystals, we can identify a particular herb. Qualitative microscopy, by looking at the type of starch grain. Large starch grains, oval is potato, maize, and this is rice starch. This is simple starch, this is compound starch. You have too many grains which occur as agglomerates. Apart from macroscopic, microscopic, we can carry out physicochemical evaluation. Moisture content of a drug will help us identify whether the herb meets some standards or no. Total ash value. Total ash value tells us whether a drug is adulterated with inorganic ash. So, sometimes what happens when we collect the rhizomes from beneath the ground, suppose you see ginger immediately after the rainy season or during the rainy season, look at sweet potato now which is there in the market. It comes with lot of earth when these are collected. I am giving simple example so you can correlate. This comes with lot of adhering mud or earth to it. So, when these drugs are dried as such and powdered, then the ash value goes very high when the drug is incinerated. Suppose during watering of the plants, if lot of mud is splashed on the leaves and leaves are dried with that mud, and when you incinerate this drug, the mud remains unburned, or that is the inorganic part due to which the ash value goes very high. So, by simply burning or incinerating the drug in a muffle furnace, we can find out whether the drug is adulterated with silica or earth. The ash value should not be very high, and these are different types of ash values, which water soluble ash tells us the total amount of constituents present. Acid insoluble ash tells us the total amount of inherent inorganic matter like calcium oxalate, whether it is present in the drug, and extractive value tells us how much the drug is yielding to a particular solvent. So, this also tells us whether the drug has required amount of active constituents. So, these are mentioned, these are official values mentioned in different official texts, which by comparing will help us authenticate a particular drug. For an organic matter, unwanted matter which is present, suppose we are collecting leaves and too much of the stem is present, then we say that there is too much of foreign organic matter. The official drugs says that the active constituent is present in a particular plant part. Other than that plant part, if anything else is present, then we consider that as foreign organic matter, and each official standard mentions a particular value where it should not exceed that value. The volatile oil content, swelling index for drugs like isabgol and tragacanth, when they absorb water, the amount of mucilage present can be calculated. Foaming index, the foam which is generated because of presence of saponin, which is an active constituent in the drug, will help us identify the drug quality. Bitterness value, when a herb is used as bitter, this is by tasting. A panel is appointed and different dilutions of drugs are made to be tasted by this panel. Tannin content can be found out by either titrimetry or spectroscopy. Very important tool in identifying or authenticating a drug is chromatographic and spectroscopic methods or techniques, which has revolutionized the herbal authentication by enabling creation of a unique chemical fingerprint for each plant. So, these methods will help us identify a constituent from a complex mixture of secondary metabolites that characterize botanical materials. So, different types of chromatographic systems are present. The commonly used on a lab scale is thin layer chromatography or high performance thin layer chromatography or high performance liquid chromatography, gas chromatography, column chromatography, paper chromatography for amino acids, and the mass chromatography, which is used to identify active constituents based on their molecular weight, and if we want to identify some liquid volatile constituents, we can use the gas chromatography. TLC is the simplest method to identify an active constituent whether it is present in an herb or no. So, here what we do is we use a stationary and a mobile phase. For a simple use, this is a silica aluminum plate coated with an aluminum plate coated with silica, and we apply the standard compound and an extract to be analyzed. Now, suppose we have a tannin extract, an extract of say harda, beheda, and we want to find out whether it contains tannins, whether it matches the tannin content. We can take gallic acid as a standard here, and you apply any tannin containing extract. This plate is dipped in a mobile phase, and the mobile phase rises due to capillary action. Once the mobile phase reaches this height, the plate is removed from the mobile phase, dried, and it is observed under UV light. You will see that gallic acid standard has run, and you will see a spot here. If this extract also you find a spot here, we can match the RF values and we can identify saying that gallic acid may be present in that extract, and hence it can be considered authentic. Likewise, each herb has got different marker compounds, which we will discuss in detail in the standardization topic. HPTLC is this is an automated machine which is used for application and for detection, but the principle is same as that of thin layer chromatography. HPLC is high performance liquid chromatography, where there is a column and the compound is eluted from the column. So, this depends on the affinity of the compound towards the silica present in the column, and the affinity will decide the eluting time of the compound from the column, and these retention times will help us identify different compounds. This is liquid chromatography mass spectroscopy, which is an advanced technique, and it combines high performance liquid chromatography with the identification power of mass spectroscopy, and the molecular ion peak obtained here by matching this with the test compound, we can identify different kind of active constituents present. Gas chromatography, this is another analytical technique which combines the separation power of gas chromatography with mass spectroscopy. These are different techniques used for identification and authentication. A very important modern day authentication technique is based on the DNA fingerprinting, like how we have the human genome and different genetic sequences. Each plant has got a genetic code, and by identifying a particular genetic sequence present, we can authenticate a plant. For example, if we want to identify a cinchona plant, we can do a DNA fingerprinting of cinchona extract and identify the DNA sequence for quinine, and thus we can say that the particular plant is of cinchona. Thus, in conclusion, the identification and authentication of herbal material forms the foundation of any quality assurance of a herbal medicine. Through the integration of classical techniques and the age old techniques like macroscopy, microscopy, combining them with chromatographic and spectroscopic techniques and the molecular tools like DNA fingerprinting, we can ensure purity, safety and efficacy of herbal formulation. These methods not only preserve the traditional knowledge, but also strengthen scientific credibility in a herbal product development, and we must remember that Authenticity is the first step towards safety, and the science is to bridge that connects the tradition to trust. Thank you.